Welcome everyone who's in the room and also online. Thanks for joining us today. I'm Phoebe Zarnetsky. I am Hazel's master's thesis advisor. And I'm just going to do a really brief introduction for Hazel's talk, and then we're going to turn it over to her and hear all about the great work she's been doing. Before she started this master's degree, Hazel was also at MSU and did an undergraduate degree in environmental biology and plant biology with a minor in environmental and sustainability studies. And throughout her time at MSU, Hazel's been really involved in outreach and engagement, especially working with the MSU Science Festival. She's been a really integral part of helping organize that festival, which happens every year, as you know, and many of us participate in. So thanks for all your hard work with that as well. And with that, I'm just going to introduce Hazel's talk here and turn it over. So she's going to talk about investigating the scale-dependent relationships of plant-frugivore biodiversity using big data in a biodiversity hotspot.
Awesome. Thank you. Today I'm going to be talking about my research, just a little outline. I'm going to go through the background, research question, methods, results, and discussion. Starting with the background, these are biodiversity stripes that visualize the change in global biodiversity over time from 1970 to 2018. The highest level of biodiversity is bright green on the left at 1970, and we can see moving from left to right, the green becomes yellow and then gray, indicating lower levels of biodiversity. On the right side, you can see the gray, the darkest gray indicates the greatest decline at 2018. As illustrated in those biodiversity stripes, biodiversity is rapidly declining and has been quantified by numerous studies. One estimates that 24 to 55% of biodiversity will be lost by 2100 if the current trends continue. If we want to prevent the worst case scenario for biodiversity loss, by increasing conservation efforts, we must look at what's driving it. The causes of biodiversity decline are well known: climate change, direct exploitation, invasive species, land use change, pollution, among other factors. We also know that these factors impact components of biodiversity differently, as shown in this figure. For example, climate change may not impact species population as much as it impacts species traits. In contrast, land use change has a larger impact on species populations than species traits. The scale at which ecological processes occur also impacts their outcomes. For example, this figure illustrates that climate impacts at fine to smaller scales, as well as largest and the coarsest scales. In contrast, species interactions have the greatest impact in scales ranging from the plot size to around the country size. In order to predict future changes, we need to understand current patterns of biodiversity. Historically, that has been focusing on taxonomic diversity. Taxonomic diversity is often measured as species richness or the number of species in an area. For example, this map illustrates global plant species richness. However, taxonomic diversity doesn't take into account the function or role that species play in a given ecosystem. Functional diversity, by definition, does, as it's a measure of the diversity of traits in an ecosystem. Some areas might have a lot of unique species with distinct functional traits or roles, where the loss of a few unique species performing their unique roles might disrupt ecosystem functioning. The figure on the right is a conceptual diagram of how the diversity of functional traits determines ecosystem function and how some of the expected impacts of global change drivers such as extinctions may impact these processes. Since taxonomic diversity or species richness and functional diversity measure different aspects of biodiversity, they are not always aligned. We can look at this misalignment of those diversities by comparing them directly. This figure is from a study that investigated the relationship of species richness with 3 different functional diversity metrics for vegetation across the successional gradient of the Atlantic forest in southern Brazil. It found that all dimensions of functional diversity increased with species richness, but differed by how much depending on the metric used. We also can investigate the interdependence of interacting species by looking at the relationships between their diversity. In this study by Delling et al., they looked at the relationships of species richness and functional richness of fruiting plants and frugivorous birds in a reserve in Peru. They found a positive relationship between plant species richness and bird functional richness, as well as plant functional richness with bird species richness and functional richness. The strongest relationship was between plant functional richness and bird functional richness. Patterns of biodiversity also vary by scale. Scale is made up of two components, grain or resolution and extent. Today I'll be focusing on grain. On the left are maps of amphibian richness at varying spatial grains. Green is highest richness, then yellow to orange, and white is lowest richness. As you can see, the pattern of richness changes as grain is increased or coarsened. We expect that as the plot size, or in macroecology, the plot size is spatial grain. As we coarsen that spatial grain, or that plot size becomes larger, the number of species also increases. Another example can be seen in this map of global vascular plant species richness at various spatial grains. Across all of these different regions, we can see a positive relationship between the log grain size and the log richness, with some regional variation in relationship strength. Functional diversity can also vary by spatial grain. It is generally expected that finer grains will have the highest functional diversity and coarser grains will have lower functional diversity due to finer grains more likely to capture the microhabitat variation in species interactions, while coarser grains integrate landscape level processes and species turnover. This pattern can be based on the specific ecological setting, metrics used, and traits. One pair of interacting species is fruiting plants and fruit-eating birds and mammals or frugivores. They have mutualistic consumptive and seed dispersal interactions. Fruiting plants provide essential food for frugivores. In turn, frugivores help maintain plant species diversity through seed dispersal. And numerous studies like this one shown here have examined this plant-frugivore interaction as networks. This example here is from the Andes, which features networks from various elevational ranges. And as you can see, there's different amounts of lines indicating the differences between specialists and generalists of plants and frugivore species. We can also look at the mutualistic relationship through trait matching, or when traits of interacting texts are compared. Trait matching is often used to better understand the interconnectedness and interdependence of species in an ecosystem. This figure is an example of trait matching for fruiting plants and frugivores by looking at gape size or mouth size and fruit size. This study quantified the associations between palm fruit size and bird gape size across the globe. Each point is one of the countries in the study, and the color is the region of that country. They found that fruit and gape size are positively correlated at the global scale. To investigate these interactions at various spatial grains, we can use functional diversity, which will help us understand the functional linkage between patterns of biodiversity for plants and frugivores. The tropical Andes is an important biodiversity hotspot that covers more than 158 million hectares in South America in the countries of Venezuela, Colombia, Ecuador, Peru, and Bolivia. It's home to 45,000 species of plants, 20,000 of which are endemic to that region, and around 4,000 species of vertebrates, half of which are also endemic to that region. It also contains 429 key biodiversity areas and more than 130 ecosystems. The map on the right shows the tropical Andes hotspot in gray with the key biodiversity areas highlighted in green. Tropical Andes is also of elevated conservation concern as as the area is warming faster than anywhere else outside the Arctic and is expected to experience some of the most drastic change in climate in South America to things like temperature, precipitation, and seasonality, illustrated by the figure on the left. These changes will also be highly variable due to the various elevation range in this region, as illustrated by the figure on the right. To better understand patterns of biodiversity for conservation in a biodiversity hotspot at elevated risk for global change, we need a more complete knowledge of the relationships of fruiting plant frugivore diversity for different metrics, and also how the relationships of fruiting plant frugivore diversity for different metrics change across spatial grains. So this study focused on three main research questions. First, what spatial grains are fruiting plants frugivore taxonomic diversity and fruiting plant frugivore functional diversity most aligned or highly correlated? And we expect that the greatest alignment would be found at coarser grains, as more variability is captured at finer grains and generalized as grain coarsens. Also, environmental or habitat heterogeneity and low rates of species turnover is more likely at broader grains. Secondly, what is the difference between the strength of fruiting plant frugivore taxonomic diversity relationships and fruiting plant frugivore functional diversity relationships? And we expect that the alignment between the functional diversity relationships will be stronger than the taxonomic diversity relationships, as mutualistic interactions and trait matching are better captured through functional diversity instead of simply species co-occurrence that may be captured through taxonomic diversity. Thirdly, what is the difference between the scaling relationship of fruiting plant frugivore taxonomic diversity and fruiting plant frugivore functional diversity? And we expect that the fruiting plant functional diversity scaling relationship will be weaker than the taxonomic diversity scaling relationship, as taxonomic diversity is expected to increase as scale broadens due to the species area relationship, whereas functional diversity is expected to experience trait clustering and functional redundancy as scale broadens. So now I'll describe the methods used for this study. So the study region, forests are an important habitat for conservation consideration, especially in the tropical Andes, as they're home to 80% of the world's terrestrial biodiversity. Also, tropical Andes forests are categorized as critically endangered by the IUCN, or the International Union for Conservation of Nature. And finally, frugivory is essential to maintaining forest structure. So for the study taxa, fruiting plants, here's just some of the species included in the study. These are pictured are some of the species with the most occurrence are pictured here. And the images are from iNaturalist users in tropical Andes countries. So for fruiting plants, we included 3,128 species. And then for frugivores, same thing. The pictures are from the most abundant species, and they're from iNaturalist in tropical Andean countries. We included 1,074 species of frugivores, and we defined frugivores as bird and mammal species with frugivorous diets above 10%. So the project workflow follows the environmental data initiative's thematic standardization of L0 being raw data, L1 modifying and cleaning the data, and then L2 analyzing data. It also aligns with FAIR data principles of findable, accessible, interoperable, and reusable data. And all of the workflow steps were conducted in R using RStudio. There are several different data sources we used in the study. For occurrence records, those were obtained from GBIF, or the Global Biodiversity Information Facility. For plant traits, we obtained records from the Botanical Information and Ecology Network, or BN, the Global Inventory of Floras and Traits, GIFT, and also the TRI database. And then for frugivore traits, we used the Frugivoria database, which is led by Beth Gernster, a collaborator and former Space Lab member. So for the workflow, again, L1 was data acquisition, and then L0 was data acquisition. L1 is data subsetting, cleaning, standardization, and then L2 is the data analysis. We used occurrence records for taxonomic diversity, and trait records are used for functional diversity. So for L0, for occurrence data, we got a habitat designation subset from the IUCN for the forest habitats, and then we created a tropical Andes forest shape, used that to get occurrence records from GBIF for both frugivores and plants. And then for trait data, as mentioned, pulled plant traits from BN, GIFT, and TRI, and then frugivores from Frugivoria. The traits used for plants were plant height, fruit mass, dispersal syndrome, plant lifespan, fruit type, seed mass, seed length, seed width, growth form, and the frugivore traits were body size, body mass, diet categories, and generation time. And these traits were selected due to their ecological importance for the interactions between plants and frugivores, and also additional plant traits related to dispersals. The trait values we collected describe species level measurements and don't account for interspecific variation. For L1, for the plants, we cleaned the raw occurrence data to remove questionable records. Since the traits, plant traits came from multiple databases, we had to taxonomically harmonize those names, the species names, so we cleaned those. And then the plants were subset to species with fruiting trait information. The traits were then standardized to get one value per species, and we used imputation to fill gaps in species trait information, imputing to the family and genus, with genus being preferred over family, and then plant occurrences were subset by species with full trait coverage. Similarly, for frugivores, the occurrence data was cleaned. We subset the traits to the traits of interest, and then frugivores occurrences were subset by species with full trait coverage. The final occurrence and trait data for both plants and frugivores was used to create diversity input objects, such as species assemblage matrices and species trait matrices for each spatial grain. So this is just showing the data amounts at various steps for plants and frugivores. So on the top, the L1 raw data, amounts of records, species, genera, and family, and then after the occurrence cleaning, and then the records with full trait coverage. And then here is just a visualization of the occurrence points in the tropical Andes for both plants and frugivores. For our analysis, we used these various spatial grains. So our diversity measures were mapped at 5, 10, 25, 50, 75, and 100 kilometers, resampling the data for each spatial grain. For our data analysis, we looked at taxonomic diversity, functional diversity using the MFD package, and then diversity relationships using the GG trendline package. And then there's images of other R packages used through the workflow. For diversity, taxonomic diversity was quantified as species richness. For our functional diversity metric, we chose to use functional dispersion, which measures the average distance of species in trait space from the centroid of the community. This metric is not influenced by species richness, unlike other commonly used metrics, such as functional richness, so we can ensure that our functional diversity is not influenced by richness. To quantify the diversity relationships, we used Pearson's coefficient R squared values, and then we also looked at the slope coefficients to look at the increase in the number of standard deviations in frugivore diversity, with one species increase in the standard deviation of fruiting plant diversity. And we looked at those for fruiting plant richness and frugivore richness, and then also fruiting plant functional dispersion and frugivore functional dispersion. And for each relationship, we fit trend lines using the two best models. So now I'm going to talk about some results. So here are richness maps across the various spatial grains and comparing plants and frugivores. Just zooming into a couple spatial grains for plants and frugivores, just looking at the spatial agreement and disagreement between the frugivore 25 kilometer and 100 kilometer, we can see that there's similar patterns, but also we capture more variability in the smaller grain, the finer grains. We can also compare the agreement between frugivore richness at 25 kilometers and plant richness at 25 kilometers. And we can see that there is also some agreement, while also some differences. And we can investigate those relationships using scatter plots, plotting the values of fruiting plant richness and frugivore richness for the corresponding cells at each spatial grain. The dots have a transparency to see the overlap of multiple points. So the darker points means there's more than one there. And going at the different spatial grains, we can see that there's a different the relationship between plant richness and frugivore richness changes as we course in the spatial grain. Similarly, for functional dispersion, maps of functional dispersion at the various spatial grains for plants and frugivores. And looking at the spatial agreement and disagreement between these maps for the various grains and plants and frugivores, we can see similar patterns that between spatial grains of the same taxa, there's some agreement, but also disagreement. And then across taxa at the same spatial grain, we can see some indication of agreements spatially. Again, plotting the values of fruiting plant functional dispersion and frugivore functional dispersion for the corresponding cells at each spatial grain. We can see that these relationships differ from the richness relationships. There's a lot more clustering happening, and it's not as linear in most instances. So comparing the spatial grains and the relationship to answer our first research question, what spatial grains are fruiting plant frugivore taxonomic diversity and fruiting plant frugivore functional diversity most aligned? In both taxonomic and functional diversity, they are most aligned at the coarsest grain of 100 kilometers, which is what we expected. For our second research question, looking at the difference between the strengths of the relationships between taxonomic and functional, fruiting plant frugivore taxonomic diversity relationship was stronger across all spatial grains for both models, which was contrary to what we expected to see. For the relationship between fruiting plant and frugivore richness, we can see that the R² value increases as spatial grain increases for both the linear and exponential model. And when we look at the slope coefficient, the linear model has a higher slope coefficient at all spatial grains compared to the exponential model. Finally, for functional dispersion, looking at the relationship between fruiting plant and frugivore, we can see that the R² value increases as spatial grain increases for both models. And then looking at the slope coefficients, the quadratic model had a higher slope coefficient at most spatial grains, but the difference between the two might not be significant. So to answer the third research question, the difference between the scaling relationships, we can see that the R-squared value was higher for taxonomic diversity relationships across all spatial grains for both linear models for functional and taxonomic diversity. And then when we look at the slope coefficient, it was also higher for taxonomic diversity across most spatial grains when we compare it to functional diversity. So overall, the taxonomic diversity relationship had a stronger scaling relationship than functional diversity, which also did not match our expectations. Now, the discussion. So some reasons that we might have seen that might explain the relationship for both plant-frugivore taxonomic and functional diversity being strongest at the coarsest spatial grain is sampling effect. We often see that more data is, we know that more data is considered at larger grains, which impacts our measurements. Environmental and habitat heterogeneity also impacts these metrics, with environmental variability increasing as grain coarsens. And species turnover. Smaller areas tend to have higher rates of species turnover than larger areas, which also could be impacting this relationship. We also found that fruiting plant frugivore taxonomic diversity relationships were stronger across all spatial grains. And this could be due to functional redundancy, the occurrence in the same ecosystem of species filling a similar role. So functional diversity might not be as high due to that redundancy. And trait selection, the traits that we use may not be specific enough to frugivory to capture that high degree of trait matching between fruiting plants and frugivores. Our third finding, that fruiting plant frugivore taxonomic diversity had a stronger scaling relationship than fruiting plant frugivore functional diversity. Taxonomic diversity may be better at capturing habitat availability, connectivity, and dispersal abilities. And functional diversity may be weakened by trait plasticity or the variation between species, and trait clustering, where species in a community share similar functional traits. Some other factors that may be causing these patterns is gap in trait coverage. Between plant trait data and vertebrate trait data, there is a lot less taxonomic coverage, as illustrated in this top figure. Even across all databases, there's still a lot of taxa that don't have a high percentage of trait data. There's also a lot more undescribed flowering plants than birds and mammals, likely due to the sheer number of species, but also the lack of focus on plants compared to birds and mammals when we're considering conservation. We also, there's also a lack of coverage in specific morphological traits. So other studies have identified traits when studying fruiting plants and frugivores that are more specific to frugivory, which could lead to a stronger functional relationship. So for frugivores, things like gape size, bill width and length, and also wing shape. For plants, more fruit specific traits like fruit length, width, diameter, shape, color, and others. In conclusion, these findings can contribute to our understanding of how fruiting plant frugivore interdependence impacts the relationship of how their taxonomic and functional diversity across spatial grains to better inform conservation. And thinking about conservation implications, using current patterns of biodiversity for fruiting plants and frugivores can help us better understand the implications of global change, such as climate change, that threatens biodiversity and ecological interactions. We also know that species extinction rates are grain dependent, and many frugivores are facing extinction, and their extinction could lead to mutualism disruption and ultimately the co-extinction of their specialist fruiting plant species, which would have functional consequences for tropical forest. So some takeaways, fruiting plant frugivore taxonomic diversity relationships and fruiting plant frugivore functional diversity relationships vary by spatial grain, and more frugivority specific traits for fruiting plants and frugivores are needed to more completely understand how these relationships exist across space in order to inform conservation efforts. And I'd like to end by acknowledging my collaborators, Dr. Phoebe Zarnetsky, Dr. Beth Gernster, and Pat Bills, and also the Spatial and Community Ecology Lab here at MSU. And I'd also like to acknowledge members of my committee, Lars, Kyla, and Nick, and then also my funding sources. Yeah. So the question was, how applicable are these findings to other regions? I would say that I think it's a lot of context dependence. What might be true in one ecosystem might not be true in another. Depending, like, there's just so much variability that could impact all these relationships. And there might be differences in data availability across different regions that, you know, could affect these patterns as well. So the methods might be applicable, but I don't think the findings can really be applied elsewhere.
Can you give an example, like Apple, you talked about herbs that have flash.
Yeah.
Like, what would cause that? I guess we have to talk about why we have to talk about the parts.
Yeah. Well, I think elevational differences and like the degree of specialist versus generalist species in the area could impact the relationships. And I think especially in tropical ecosystems, a lot of plants are known to be involved in frugivority interactions. So maybe in other regions that might not hold true. Yeah. Yep. So the question was talking about the sampling differences between plants and frugivores, especially at the smaller scales. And I'd say there probably is likely a big difference. You know, birds and mammals are easier to see. A lot of more study has been done for them. There's less species to look for and document. Whereas like the sheer number of plant species, it's just hard to capture all of that. I also, there might be some similar like sampling bias occurring between the two, just like accessibility. You know, some areas may be hard to access. Yeah, definitely varied.
Or animal species.
The question was, do I have a favorite plant or animal species in the database? And I actually didn't look at them on the individual level until I was picking out pictures for the presentation. So I'm gonna say no. I'm a generalist, not picking favorites. Yeah. Yeah.
But do you have a sense of how many, or what the breakdown was in terms of generalist versus specialist relationships between the plants and the frugivores? Yeah, so the question was, thinking about the differences, the breakdown between specialist and generalist in the data set, and I don't really have a good idea of the difference between them, just based on the sheer number of species. I think that's easier to look at when you're looking at the networks and like pairwise interactions versus like across the broader scales. Yes.
Your results of the
Mm-hmm.
given limitations of those databases. Is there anything you might?
Yeah, so the question was about the data limitations for traits and, you know, how the functional diversity findings didn't align with our expectations. So what I would do differently to maybe try and get at that at a different angle. I think maybe looking at a different metric of species or functional diversity maybe could help. Also maybe expanding the frugivore trait data to include, I think there's been a couple new trait databases out in the last like year or so. So maybe those could fill some gaps in for that, for the like more specific traits.
If you filter out a link for a straight link for your head bar, did you?
For the frugivore traits, the traits were limited to the traits that were most related to frugivory and also had information for both birds and mammals. Yeah.
Do you think you would find something different if you separated out birds and did this analysis versus mammals?
Yeah, so the question was, do I think I'd find something different if I separated out the birds and mammals? I hadn't really thought about that, but I did think about, we defined frugivores as like birds or mammals with like 10% frugivorous diet. I think maybe if we increase that to like maybe like 50%, I've seen in other studies, that might help capture like those relationships better.
For coming.